So yeah, it's the Z lines because the Z lines indicate the length of our sarcomere. And if our muscle contracts, that means our sarcomeres are getting shorter. So therefore the Z lines are moving closer together. A, H, and I are incorrect because those aren't lines, those are kind of areas of the muscle. So those are different bands. So ATP is kind of loading it back. And then once it's loaded back, it essentially gets hydrolyzed during then. So it turns into your ADP and P, and that's kind of loaded back. And then during crossbridge cycling, it swings across, and then ATP once again reattaches, cocks it back. So the role of ATP there is to load the myosin head into a cocked position. So distal stimuluses are the things that produce the sound, whereas the proximal ones are the things that are actually reaching you. So the phone speakers are the distal stimulus because that's where the sound is kind of coming out of. And then the actual sound waves are what's going into your ear and hitting your eardrum. So therefore it's the proximal stimulus. Afferent and efferent we don't really use too much. Those we mostly use for sensory and motor signals, where sensory is afferent and motor is efferent. Wait, shit, did I get this wrong? Heine? The Babinski response. It's when it fans out, right?
Yeah.
Okay, I will come back to this one. Yeah, good. So teres minor strength, or the teres minor in general, is for external rotation of the shoulder. So therefore we test it in clinical skills by getting the patient to externally rotate their shoulder while kind of pushing against them. And then we can rate that from a scale of 0 to 5.
And yes, Babinski is the toes will fan out. So when you stroke it, it's supposed to go like that.
I think there was a bit of confusion with the question. I think they were confusing it with Babinski's reflex. So
your mic's like broken.
In adults, when you do it, it should be that we scrunch our toes. So if in an adult you see the toes fan out, then you know there's an underlying problem there. But if we're testing it in a newborn infant reflex, then it should be fanning out.
Yep. So Babinski reflex is normal in children. In babies, sorry. Yeah, so this one is talking about the carpal tunnel, which goes through your wrist, mostly important for carpal tunnel syndrome, where you get compression of the median nerve. So I'm glad no one picked the median nerve. That's a good sign. I believe with the carpal tunnel, you can draw a cross section of it, and you essentially have, I think it's 4 tendons for the flexor digitorum profundus, and then 4 for the superficialis. And then the flexor carpi radialis doesn't actually pass through the carpal tunnel. So therefore, it's also not affected by carpal tunnel syndrome. Yep, so with this one here, we have either the lat dorsi or the trapezius. And trapezius is the correct answer here. Trapezius is supplied by one of your cranial nerves, which you'll learn next year. That cranial nerve is the accessory nerve, and that also does the sternocleidomastoid, which is on your neck. Whereas your lat dorsi is supplied by lat dorsi. Let me Google it. Lat dorsi is done by the long subscapular nerve. So not quite from the, it's still from the brachial plexus, whereas the spinal accessory isn't. Biceps brachii and brachioradialis, but the biceps is innervated by musculocutaneous, whereas the brachioradialis is probably from your median nerve, I think. In other words, from the brachial plexus. So yeah, 60 to 120, or in other words, the middle third of 0 to 180. So painful arc, we're doing shoulder abduction. And the only muscle here that's responsible for abduction is the supraspinatus, because it kind of gets caught underneath some of your bones there. Whereas subscapularis is for shoulder internal rotation. So we're not testing that with painful arc. Yeah, pharmacodynamics is what the drug does to the body. So how does the drug help us or harm us? Whereas pharmacokinetics is what the body does to the drug. So how we get rid of the drug, how we metabolize it. Yeah, and pharmacokinetics would include our blue and our green options as well. So therefore pharmacodynamics is what the drug does to the body. So IV drugs are 100% bioavailable because the definition for bioavailability is an IV drug. So it's always going to be 100% there.
This is a high yield question.
So this one, yeah, most of the time we pee out like excess drugs, and that occurs in the liver. So our drugs are metabolized within the liver mostly, and that serves to make it more water soluble so that our kidneys can process it, and then after that we can pee it out. So therefore drugs are made more water soluble during metabolism. And that occurs in the liver, but it can't be liver soluble, if that makes sense. Yeah, glad that everyone got this right. So it takes 4 to 5 to achieve steady state, as well as to also eliminate. Yeah, that's good. So zero order elimination, yeah, happens when they're saturated. So that means that the rate of elimination is essentially constant. It's linear because no matter how much drug you have, the enzymes are working at their full capacity. So yeah, it's all to do with enzyme saturation for zero order and first order elimination. Yeah, pili and fimbriae are kind of like the sticky bits on the outside of the pathogen, and the pathogen tries to stick onto our cells. So yeah, so that's so it can like penetrate or it can continue spreading. Whereas the ones to avoid phagocytosis, that one is typically our capsule. So our like our sugar capsule that kind of hides our pathogen from our immune system. Okay, so H. pylori or Helicobacter pylori, yeah, really high yield, I think, especially for GIT next year. So yeah, it doesn't actually neutralize the stomach acid. Most of the time when it causes stomach ulcers, it's essentially because it gets rid of that protective layer on our stomach. And if it gets rid of that protective layer, then our own stomach acid is the one going to be attacking our stomach lining. So therefore, urease is what digests it. Yeah. trigger mechanism and zipper mechanism are both ways for them to enter. The zipper is literally like a zip you would find on like a piece of clothing, and that happens with the cell, so it can enter it. Trigger is very similar, except instead of it being zipped up, it kind of just plunges straight into the cell. Slot is just not a thing. Does anyone have any questions about anything there? Otherwise, we'll probably get started. Week 4. Otherwise, good job, everyone.
Jiting, do you want to go first?
Yeah, I'll go first. My slides are first anyways. Just let me share screen. Okay. So going through Bacterial Pathogenesis III, which I'm pretty sure is the last one in your series of Bacterial Pathogenesis lectures. Okay, so in this one, we're going to focus on how the bacteria actually causes damage to the host. So I'm sure in previous lectures, you would have gone through this whole infectious cycle. So now we're essentially at the stage of the bacteria is now in our body, it's colonized it, but how does it actually damage the host to cause the symptoms and signs of disease that we use to then diagnose the patient? So the main thing we're going to look at today is the fact that bacteria can essentially release toxins. These toxins can vary in terms of their mode of action and can range in the severity from minor to being very deadly. The two main classes of toxins, you might have heard of this, is endotoxins versus exotoxins. Another way for bacteria to cause damage is its metabolic byproducts, because some of these byproducts can also cause damage to the body. Another one is the host or our own immune response actually causes the primary damage, and this is typically in the form of a huge inflammatory response that overwhelms us. So let's go through the types of bacterial toxins first, so endotoxins versus exotoxins. So endotoxins, it's actually an LPS component, or the lipopolysaccharide component of the outer membrane of gram-negative bacteria. And it's only released when the bacteria actually dies. And since it's an LPS component, it's only present in gram-negative bacteria. The active component, however, of the LPS that makes the endotoxin a toxin is the lipid A component, which you can see in this diagram here. Exotoxins, based off its name, it's just essentially toxins that are secreted actively whilst the bacteria is still alive. Okay, so here are a few definitions for sepsis, hypotension, and septic shock. So sepsis is referred to as the whole body's systemic inflammatory response in response to a microbial infection. Hypotension is just the word for low blood pressure. And septic shock is sepsis-induced hypotension that would normally persist despite adequate fluid treatment. This can lead to multiple organ failure because we have insufficient blood reaching our vital organs. And essentially the take-home message is probably that sepsis is a medical emergency, and it can lead to death very rapidly if we don't do anything about it. So how does someone actually get sepsis or septic shock? So the source is commonly systemic infections caused by gram-negative bacteria. So the effects of gram-negative bacteria in terms of how they cause septic shock is indirect. It's normally mediated by host molecules. So if we look at this diagram here, so how does the endotoxin or the LPS of gram-negative actually lead to shock? It's not LPS directly causes shock straight away. LPS essentially does a lot of things such as initiating cytokine release from our immune system, activating clotting and other complement pathways. And so all these things together will then indirectly cause septic shock. So we're also going to go through the major classes of bacterial toxins, and they're separated into classes based on their mode of action. So we're going to go through three main ones today. The first one is what's called super antigens, and these are toxins that activate our T cells. The other type are toxins that act at the to target the cell surface. Another one are toxins that modify intracellular targets. So these are the three we're going to go through today. So let's start off with super antigens, which I said before, they activate T cells. So these are bacterial antigens that are secreted actively by the bacteria. They non-specifically bind to our MHC2 and TCR to activate our T cells. And so normally, with a bacteria that doesn't secrete super antigen, so let's just say a normal antigen would only activate around 0.001% of our T cells. So we have an adequate response to be able to get rid of the pathogen. However, what super antigens do is they actually activate a lot more T cells. In fact, they can activate up to 20 to 30% of our T cells. Because of this strong binding that they do here, as you can see in the diagram. This can be life-threatening because it can lead to hypotension, cytokine storms, toxic shock, and multiple organ failure because we suddenly have that huge activation of our T cells. And some bacteria that produce super antigens include your Strep pyogenes and your Staph aureus. So these super antigens is actually what a lot of people know as toxic shock syndrome, which is typically caused by Staph aureus. It's commonly associated with tampon use. And so the symptoms and presentation of this could include things minor, such as a fever, vomiting, diarrhea, muscle pains, hypotension. However, it can lead to heart and renal failure in severe cases, which then causes multiple organ failure and ultimately leads to death. Okay, so moving on to the next class of toxins. These are toxins that act at the target the cell surface. Sorry, bad grammar. One type of toxins that act on the cell surface are phospholipases. So these do hydrolysis and they break down the membrane phospholipids essentially. And so one type of phospholipase is the alpha toxin that is released by C. perfringens. And so what it does is it hydrolyzes the phosphatidylcholine and sphingomyelin, which are all components of the human cell membrane, essentially. So just know that they break down the human cell membrane. And this is also the toxin that plays an essential role in what's called gas gangrene, or the disease that perfringens causes. So another type of toxin that acts on the cell surface are pore-forming cytolysins. You can probably base it off their name, pore-forming. They form pores in our cell membrane, therefore they disrupt the membrane integrity. We have small pore-forming toxins and large pore-forming toxins. So the small ones, they just punch small holes. One is the alpha-hemolysin toxin produced by Staph aureus. And what the large ones do is they bind to cholesterol in the cell membrane and polymerize to form large pores, essentially. Okay, so moving on to toxins that now modify our intracellular target. So they don't just work on the cell surface, they actually get inside our cells. So this is the most common type of bacterial toxin, and normally these toxins have what's called an A-B structure. They have two components, an A component and a B component. So if we look at the A component, so this is the component that actually has the enzymatic activity. So this is the part that can actually modify a specific intracellular target within our cells. The B component is responsible for initially binding the toxin to our cell membrane so that our cell essentially uptakes this token or endocytoses it. So you have two distinct groups, simple AB toxins, an example being diphtheria, and compound AB toxins, being the cholera toxin. So as you can see in this diagram, we have the A and we have the B component. The B is what binds to our cell membrane, so that our cell membrane then takes up the toxin. And then A will essentially say, thank you, B, you've done your work now, I don't need you anymore. And so A will detach from B, and the A component has the enzymatic activity, and it will then go on to modify a specific intracellular target. So if we have a look at the toxins that we just mentioned, one being the diphtheria toxin. So the diphtheria toxin is an ADP ribosyl transferase. Its B component has essentially evolved to be able to bind to a part of our receptor. And what it essentially does is it can target our ribosomes, namely the elongation factor 2 of our ribosome. And what it does, given its name, it's an ADP ribosyl transferase, it'll add an ADP ribose to our elongation factor 2. And essentially when it does that, once our elongation factor 2 is modified, it's inactivated, and if our cell can't produce any proteins, then our cell essentially dies. And this is just a bit of background information of what diphtheria or the disease is, in case you guys haven't covered that yet. So the next one, or the compound AB toxin that we're talking about, was the cholera toxin. And so it has a subunit that is also an ADP ribosyl transferase. And what this toxin, or what the A component of this toxin does, is it essentially causes massive fluid electrolyte release. And this is what results in the diarrhea that we get when we are infected with cholera. And typically we see it as rice water stools, because you have diarrhea, but the diarrhea is kind of like this white murky color, kind of like what it would look like after you wash your rice. Yeah. So if we look at this diagram here, let's just quickly go through how the AB toxin works. We have the A component and with the B subunit. The B binds to the cell membrane, and this will allow the toxin to get into our cells. And then the A will essentially come off the B because it doesn't need it anymore. It's already in our cells. And so what the A component of the cholera toxin does is it will target the adenylate cyclase on the surface of our intestinal cells, and it'll essentially just tell the cell to pump out all our electrolytes. And water will follow these electrolytes out of our intestinal cells, and that's why we end up with diarrhea. In the lecture, you also touch on two main types of neurotoxins, these being the tetanus and the botulinum toxins. And since they are neurotoxins, they act on neurotransmission, or they interfere with neurotransmission. So botulinum toxin blocks the release of our neurotransmitter acetylcholine. And this will essentially result in flaccid paralysis. So you become floppy because your acetylcholine can't work on its receptor anymore. Tetanus, on the other hand, works slightly differently. So the tetanus toxin blocks the release of the inhibitory neurotransmitters that would normally go and inhibit the action of acetylcholine. So this results in muscle spasms. And so you essentially can't relax again, so you're rigid, kind of like this painting here. And so this is essentially a quick diagram summing up the two toxins. So the botulinum, as we said, normally our acetylcholine, an action potential propagates down, and once it comes to the end, our acetylcholine gets released from its vesicles, binds to the receptors on our muscle cells, and we can contract our muscles. However, once we have the toxin, our acetylcholine can't be released. No acetylcholine binds to our receptors on our muscle cell, and we essentially can't get our muscle to contract. Tetanus works a bit differently. So in tetanus, our acetylcholine, it can still come out of its vesicle, can still be released, still attaches to our receptors on our muscle cells, so our muscle cells contract. However, when it's time to essentially turn it off and inhibit the actions of acetylcholine, this is when the tetanus toxin essentially stops the normal function. So over here, we don't have any inhibitory neurotransmitters being released. Our acetylcholine continues to work. We can't relax, and so therefore we essentially have rigid paralysis. And I think that's all. Any questions?
Okay. Give us 1 sec. Okay, here we go. Cool. So I'm going to be going through study design, which is actually really important, especially for your epi assignment, which I have no idea if they've told you about that yet or not, but is actually really important for when you like analyze. That being said, a lot of this is like, I'll make this really quick because you can kind of just look at this from the slides. And a lot of this is stuff that like you would probably already have like a general overview, like general knowledge of. So basically, when people think of study design, they usually think of this pyramid. So you've got your kind of bad types of study design down here at the bottom, and the ones that we are aiming for at the top here. So obviously gold standard is the randomized control trial. And then next year, when you get into, when you're in year 2 doing evidence-based medicine, you're going to be doing a lot more on this, the very, very top one, which is meta-analysis. Yeah. So just some definitions, just so it makes it, oh my goodness, realized, yeah. Just so you guys kind of like, you know what Penny is talking about when she's going through these study designs. So a case is someone who has a disease, a control is someone who does not have the disease, and the exposure is the factor that's going to, that you're probably going to be investigating, which will cause the person to either develop or not develop the disease. The outcome is the outcome. Often basically it's the development of that actual disease that you're investigating. And obviously quantitative is, did I get that mixed up? That's supposed to be the other way around. Quantitative is numerical, qualitative is descriptive. Just ignore that for a second. Anyway, so you've got what we would call like bad studies, right? Like studies that you want to kind of avoid, study design that you want to avoid using. And they are kind of all grouped in the bottom 3, right? These are the ones that you might get asked to analyze at during the assignment, you might not. But basically these ones, like we don't want to use these because they're not exactly very good. So these can kind of be lumped into a category called descriptive studies, which is kind of like in the name, they're descriptive, right? So these are your case study, case report, or a case series, as well as in the next slide, the ecological studies. Basically, the common advantage that these descriptive studies have is that they are very good for generating hypotheses for the basis of further research. So they're actually really good for seeing whether there might be a possible link between different, like, you know, exposures, whether there is actually something that's worth investigating. So yeah, and also fast, easy, cheap, very easy to kind of just do, but you don't want to use this as a basis for like maybe like determining causality and going like, oh, because I read this ecological study, X causes Y, and therefore like I make my conclusion. And it's like, okay, maybe not, maybe you don't want to be doing that. So the ones that we do want you to actually learn, the ones that are actually good, are the ones on the top of the pyramid. So we have a few. There's a lot of information on these slides, but don't worry, you can read them slowly in your own time. But basically the first one we've got is a cross-sectional study. And so basically this is a study done where it's a snapshot of one point in time, and that measures the prevalence of disease and the presence or the absence of exposure at that same point in time. So advantages, very cheap, easy. So when you're doing the assignment, one of the factors that you're going to have to actually consider is the expense, right? You don't want to be conducting multiple studies where it's going to be wasting, like requiring lots of resources, requiring lots of money, when there potentially could be a cheaper and easier way of actually doing it. Basically, the main disadvantage though, it's actually very difficult to see whether there's actually, you can establish causality. So you can't actually establish temporality with a cross-sectional study. So this is the example that will hopefully make it make sense. So for example, if you're doing a study investigating cannabis use and depression in young people, and you're trying to find the link. So if you do potentially like a cross-sectional study of young people, you can ask questions about their lifestyle, and you include a question which assesses depression. And you find that young people who report cannabis use are more likely to be depressed. However, this also begs the question, does cannabis use itself cause depression, or do depressed young people choose to use cannabis more often than those who are not depressed? So you can't actually establish temporality, you can't establish that cause and effect. But once again, it is a good basis for you to generate possible hypotheses for you to actually do further study and further research. Then we move on to the case control study. I used to always get these really confused because this is where it gets like confusing, because everything's like called the same thing. But basically, case control studies are where you will identify individuals with the outcome that you are interested in, and then you look backwards through records or interviews. Usually you want them to be like unbiased medical records to find the possible cause or the possible exposure. So the key thing to note here is that your study direction is backwards in time. So your results will eventually be expressed in an odds ratio, which is used to approximate the association between that disease and the exposure. So this is actually really good for any rare outcomes or diseases that have a slow progression. You can choose to just study the people. So obviously you're choosing the outcome of interest, right? So you choose to study the people with the disease, and then you're looking backwards to see if you can establish and find a possible cause. I think the one that they use in the lecture is like cerebral palsy. I think that's the one that they use. And it's good for those kind of more rarer diseases. Relatively easy, pretty fast, pretty cheap. However, because you're using retrospective data, so you're going backwards in time, you can't actually establish incidence and prevalence. So that brings us to the next study design, which is a cohort study. Now, I used to get these two confused, so I'm just going to preface, please don't get these two confused. So in a cohort study, you are identifying the individual with the exposure of interest, and then you are following them to see who will actually develop the disease. So the direction is usually prospective, so like moving forward, like you go start at a point and then you like follow them through time. But it also can be a retrospective cohort study. Also, I just realized I typed this wrong. Sorry guys, this was like at like 1 a.m. in the morning. So it can also be a retrospective cohort study. So that's where you're still doing it, where you identify a population with the exposure of interest, and then you go backwards looking for unbiased documentation, seeing whether they actually you see within the unbiased documentation whether they eventually actually develop the disease. So the study design is always, for the cohort study, it's always going to be moving forwards in time. Whereas in the case control study, it's always backwards in time. You're always going backwards. So your results will eventually be expressed in a relative risk. So some advantages, it's very good for showing temporality, and it directly measures incidence. Very good for estimating the risk of actually developing a disease following a specific exposure, because you are starting with the exposure and then seeing whether it actually will develop into a disease. Very good for rare exposures as well. But however, it is expensive. One of the biggest disadvantages for cohort studies is they're hella expensive, hella time consuming. And a lot of the time when you're, especially for retrospective cohort studies, you will be following a group of people who you're like, yes, okay, this is my exposure of interest. And a lot of people will fail to actually follow up. So you'll lose a lot of documentation that you're actually looking for. So you'll have like a huge population at the start and you'll end up with like little to no data, which is always like a bummer. And then we move into the gold standard of study design. So our randomized control trial, RCT, which is kind of like what you want to do for everything. So basically advantages, you're free from known and unknown confounders because everything is in the name, randomized. And you can even do double blinding or even triple blinding. So basically neither the participants nor the experimenters will know who is receiving the particular intervention, whether that's a new drug versus placebo or a new drug versus an old drug. Some disadvantages is that sometimes the interventions that you are actually investigating are not ethical. So you can't forcibly put someone through a particular like RCT. So you can't actually do that. And yeah, that's one of the major disadvantages to a randomized control trial. And then last of all, you have your systematic reviews and meta-analyses. You won't do much of this in year one. This is very much like you'll basically like your whole assignment in year two for evidence-based medicine is a systematic review, meta-analysis. This is, however, the foundation to evidence-based medicine. So this is kind of where we want to be getting all of our data, where we want to be getting all of our conclusions to form the basis for how you practice every day. So a systematic review is basically you review all of the literature that is relevant to a particular question. And you want to be reviewing as many like high quality RCTs as possible. And basically after reviewing all of the literature, you're trying to come to a conclusion about as a response to that particular question that you're investigating. And yeah, meta-analysis is basically the same thing. You're combining the results of several different very high quality studies that address related hypotheses, and then you're trying to come up with the conclusion, coming up with statistics and overall information. And that is study design. You'll also be going to, you're also going to do like a tute on this as well, where they'll go through, especially like cohort and case control studies in a lot of detail. But yeah, that's basically it from me. Does anyone have any questions?
Cool. I think that's like everything. That's everything.
Everything we wanted to say today.
Yeah, like pretty short week. So it's good. So yeah, slides are up. Lower limb slides are also up already. Just finished them yesterday. So check them out if you really are keen, but like don't feel pressured to.
How is MSK? I've just been hearing a lot of complaints about MSK. It's like how bad it is and how boring it is.
I'll stop recording as well.